Hello everyone. Today we will see in-silico drug discovery from structure to simulation. Suppose you have some chemical compounds identified from the GCMS or LCMS or FTIR or any other analysis. So you want to check whether your compound has drug activity or not. So how you will start your in-silico study with your compound to check drug activity. So first of all, you have to identify your structure, and for optimization, you have to obtain or collect this structure. We know that chemical structure has two view: 2D structure, 3D structure. We can draw 2D structure if the compound is a novel compound, and we can draw by ChemSketch, ChemDraw software. But there are a huge database from where we can easily collect our chemical compound. Basically, we can find this chemical compound from PubChem, ChemSpider drug bank. From this web server, we can also download 3D structure. So after downloading 3D structure, you have to minimize their unusual energy, and for further study you must need to uh, minimize this energy, and also you can perform optimization by Avogadro, Gaussian software, open Babel software. And there is another things that you can also draw 3D structure by Avogadro Gaussian software. So after structure preparation or download, you must check Lipinski rules of violation. By these rules, you will identify a drug likeness property of your compounds, and from this you will get the further conception and further analysis you can determine. So by Lipinski rules of violation, we will isolate the compounds that can give a drug-like characteristics or properties. So the next step is selecting and preparing the protein target. So why this section is required? We know that our drug or our drug candidate compounds will bind to the target protein, which is involved with the disease progression or making. So we must select a protein target, and for this we have to study a vast information with the disease. We can identify the protein by several methods. We will discuss it in another video. So after selecting the protein target, you must know the PDB ID, or you can search through the protein chain name in the RCSB PDB database, from where you will uh, download the 3D structure of the protein. And after downloading the protein, you must clean or prepare the protein, as the protein chain is co-crystallized with ligands or crystallographic water and any other heteroatom. So you must need to clean these compounds or heteroatom from the protein chain, and adding polar hydrogen. Next step is energy minimization by SwissADME. So further, we need to prepare our ligand for molecular docking, and also must prepare in a format that support in the docking software. So for docking software, we need PDBQT format of both ligand and also protein. So we need to convert our any other format file into PDBQT format by AutoDock suite tools or any other docking software. So after conversion of the file format, we can also add a charge assessment, and we can define rotatable bonds. So then we will start molecular docking. For molecular docking, we can use AutoDock, Vina, PyRx, Doctor, Desmond, etc. So molecular docking precisely define the coordinates and dimension of the protein's active site, the search space or push. So run the docking simulation which sample thousands of poses and calculates predict binding energy of each ligand. After docking procedure, we must interpret our results. So first thing is interaction analysis, binding site evaluation, affinity and push assessment. Interaction analysis means identifying and categorize the types of molecular interaction, including hydrogen bonds, hydrophobic contacts, and electrostatic force at the binding site. Binding site evaluation means where our predicted ligand bind with the protein and which residue is involved in the binding procedure. So we can visualize these things with some tools or software like Discovery Studio, PyMOL, Camera, etc. Then affinity and pose assessment is required. Then molecular dynamic simulation is performed to check the stability of our ligand where the ligand bind to the active site. So it's the validation of the stability of the ligand and or the complex. So MD simulation moves beyond static snapshot to assess the temporal stability of the protein ligand complex in a near physiological environment. So purpose of MD or molecular dynamic simulation is provides dynamic view of the binding interface under fluctuating condition, temperature, pressure, solvent, and verify optimal docking pulse remains stable over time. It's maybe 100 nanosecond, 500 nanosecond, 1000 nanoseconds, and calculate the key parameters like RMSD, root mean square velocity to predict structural stability. So we can perform molecular dynamic simulation by using GROMACS, NMD, or Desmond ESR software. Then ADMET, and ADMET stand for absorption, distribution, metabolism, excretion, and toxicity. And these are very essential for filter out compounds with poor pharmacokinetic properties. And if your chemical compound has poor gastrointestinal absorption and metabolic enzyme interaction or very low excretion, we should remove this compound from our study. And also toxicity profile is a very important for choosing a compound as a drug candidate. So then proper documentation and reporting is required that your study can further help in drug discovery process.